No. Okay, good morning. I am happy to see so many distinguished people here and also young scientists. And I will try to provide my perspective on the past and the future of computational biology. I will start essentially the whole talk. The whole talk will consider the synergy and also the difference between approaches which are based on physical treatment, which is really causation, and on the use of AI, which is correlation. And I will start with physically based approach in computational biology, and also start with the issue of enzyme catalysis. Now, as all of you or most of you know, enzymes are proteins that catalyze chemical reactions. And they do it by reducing the barrier for different rate-determining steps. So understanding of enzymes require the ability to use physical models that actually describe the key processes. And again, enzymes are a large part of the protein in our body, and they do most of the critical actions. So the main approach to study how enzymes work is this combined quantum mechanical molecular mechanics approach, where we restrict the treatment of the bond breaking to quantum mechanical approach, which should not be used for the whole enzyme because they are too expensive, and describe the rest of the enzyme by classical force field, ball and springs, and electrostatic charges. Now, this approach became very effective in the last, 50 years. And just my favorite example is studying of the enzyme RAS, which catalyzes GTP hydrolysis. This is the GTP, and it activated when it bound to another protein called GAP. So, could we run this movie? And so we could use this QMMM approach to simulate the catalytic reaction. In this case, a water molecule attacked the phosphate by first transferring a proton and then by OH minus, and it get help by a magnesium ion, which is shown here in green. Now, the key challenge is actually to get the chemical barrier for the enzyme and for different mutants that could actually cause cancer. And this approach allows you to do it with a reasonable accuracy. And in fact, this approach allows you to study many enzymes. What you see here in blue are the barrier for the reaction without the enzyme. In red is the barrier with the enzyme. And in general, we found that this catalytic effect is mainly due to electrostatic stabilization of the transition state. So you could use this physically based approach to study the main catalytic effect in enzyme and to understand its origin. Now, another question which now become very popular is could you use your knowledge to design or to improve enzymes? And there are two main approaches. One is experimentally directed evolution, where you just keep choosing in solution the best enzyme in several rounds, and the other is physically based approach. Now, with physically based approach, I mean, this is directed evolution, but with physically based approach, what we do, we on the computer mutate different residues and calculate the free energy change of the reaction upon such mutation. Now, it works reasonably well. Here is the case of haloalkane halogenase, but it's not sometimes good enough. Note that when we study the whole catalytic effect, it's a very large number, so small errors do not count too much. Here we look on small effect, and we need to be relatively accurate. So in the last 10 years, we try to improve the physically based approach. The favorite system is this CAMP eliminase, which was designed from scratch and improved by directed evolution. Again, we seem to do reasonably well, but when we try to get the best mutant, the one which makes this enzyme the fastest, even the use of massive scanning, creating many, many mutants, seems to give reasonable result. The blue region is where we have the fastest mutant, but we could not get exactly the observed effect. And we still have, and we are still even struggling to try to get better result. So this is the case of physical-based effort, about 50 years. We are doing okay, but not good enough. So the question is, can we get some help from AI? And the direction, okay, first just a footnote. A long time ago, in 1975, we used coarse-grain model to try to study protein folding. We were able to show that protein could actually fold on the computer. We even got reasonable structure, but this approach and a related continuation could not produce from sequence the exact protein structure. And then come the quote-unquote surprise. Using AI, people were able to generate the AlphaFold. Essentially, it was the Nobel Prize last year, and to get very good results for protein structure prediction from sequence. Basically, to get more or less the ultimate force field. Now, this approach has a huge hype. A lot of people think that it will solve anything in biology, but actually there are many problems that it have not solved. In one of the direction, which is continuing what I told you before, is enzyme catalysis. So we tried to use an idea that was used before in studying protein fitness, to use maximum entropy and to see if it helps in creating correlation for enzyme catalysis. And just show you what happened. Okay, so just in short, maximum entropy is the idea that you look on many enzymes and you try to find the likelihood that a given mutation will appear, the probability of a mutation. So it makes a lot of sense because you look how many times each mutation appears. Now the question is what this information has to do with physical properties like catalysis. So we took different enzymes, and without pretending that we don't know anything on catalysis, we plot the calculated maximum entropy and the observed catalytic effect. Again, the approach does not have no information about catalysis, yet we found very nice correlation between maximum entropy to catalysis. These are several examples. The larger is the maximum entropy, the less is the catalytic effect. These are 3 enzymes. We found out that this works best if we look on mutants not far from the active site. This table we jump, this we jump. We also found that this approach even works for enzymes which were not designed by evolution. And very significantly, we found also that the protein stability is correlated with the maximum entropy, essentially correlated very nicely. So we have here a correlation for activity, for K cut over Km, and for stability. We are not sure why it works. Essentially, I argue with my co-worker. One possibility is that when you have the landscape of the sequence and the activity, we just stuck in a given region and we follow the trend. My co-worker believes that this is more fundamental, but again, we don't know why it works. So we could, in principle, predict activity and stability. We could find mutation that work faster than the wild type. We could find protein which are more stable than the wild type. And essentially, we demonstrate this idea by designing the halogenase which is faster than the wild type. Now, here we have a trick. It's really, really hard to compete with the whole activity of the wild type because evolution spent billion years or less to do it. But for specific steps, like the SN2 step in the halogenase, we just use our correlation and find protein, this one, which is faster than the wild type. We predicted it and then verified it experimentally. So there is something interesting that you could do with this maximum entropy. We even tried this approach on bioluminescence, where we have this cofactor luciferase that emit light after chemical reaction with oxygen. And we actually first studied the action of luciferase by this QMMM approach, and were able to understand its activity, and we plan to use this approach to find better mutant. Now, what we mean better is that it will emit farther to the blue because these creatures live in the ocean and you want blue or UV emission, so it will be not blocked by the water. So we use maximum entropy, found correlation between maximum entropy to activity, again without having any idea why this has happened. We also found, you see difference between groups which are near the active center and farther away, so it works better near the active center. We also found correlation for stability. Again, stability is correlated with maximum entropy. But the most impressive finding was that we found correlation with the wavelengths. So when you go to the blue, we know which mutant will take you farther to the blue, and this is the direction for which the enzyme evolved. And we even suggested several mutations that seems to work. Okay, so now we go to, I think, more challenging issue and try to see how and if this AI work. So the issue is drug resistance. It's a major problem. And we have been working on it for a long time with really medium or very small success. We were looking for what is called the vitality, which is based on the following. The pathogen want to not bind the drugs that we have. So it want to have large Ki. But when doing so, it also want to retain its original activity, its original catalytic effect. So it cannot have too small K cut. So what we want to monitor and to calculate is the vitality, which is the ratio between Ki time K cut over Km for the mutant to the corresponding value at the wild type. So we want to find drug that will reduce vitality. The pathogen want to have high vitality. So we were trying to calculate vitality for some system. This is HCV for different inhibitors. We calculated the binding. We calculate the catalysis. Maybe I show you the opposite slides. And from this we obtain the overall vitality for different drugs. Now, this looks nice, but it's clearly not good enough to use it for drug design. So we got stuck. One of the reasons that we got stuck, we also have no ideas which mutations the pathogen will do. What will be the next step of the pathogen? We know it wants to maximize vitality, but there are other factors in the sequence. So again, in desperation, we move to AI. This is recent paper, which probably appeared today in PNAS, where we took the huge data set of HIV protease and tried to correlate it with this maximum entropy. We got very nice correlation where you could say that the drug with the lowest vitality here is the best drug, but unfortunately, we also found out that we get the same correlation looking at the number of mutations, which means it's really meaningless correlation, though it's very nice, and you could use it only after you've done the experiment. This is expected since HIV protease has huge mutation rate. So we also done it for enzymes which is less mutated, and we found some correlation which is unique to maximum entropy and could help us if we want to simulate the step-by-step resistance. But still, no huge success. Now come, I think, one of the nicest examples, so we will try to go relatively slow. This is kinosine. It's a myosin-type system which works in one direction and carries a load. And one of the questions that we were asking in the last 7 years is why it works in one direction. And this question dramatized here. I mean, if you have two equal legs, there is no reason that you will not walk in the same speed in both directions. But in these proteins, one direction is good, the other is not good. So the question is why? And this question essentially was almost never asked and still not asked in the field. I don't know why. And after about 3 papers, we realized that what determined the directionality is not any dynamical or kinetic effect, but simply the rate-determining barrier, which appear to be different for the forward and backward direction. So when we look more carefully, this is myosin 5, we found that the one which goes forward has easier exit of ADP, which is the rate determining step, than for the ones that go backward. We published several papers on it, and recently we start to use it to medical questions. For example, there is a major heart myosin, which if it does not work, you eventually could die. And we try to look on the effect of different drugs on the rate determining step, which in this case is phosphate release. We do it by calculating the free energy barrier for the release of the phosphate. And we always find that with the drug, the rate determining step become higher. With the mutations, the rate determining step become low, and it does not, and it's not anymore rate determining step. With the drug, by the way, this blue should be orange, so always the drug helps to increase the barrier and to make it more similar to the original rate determining step. The last system that we did was the kinosines that you show in the movie. Again, it is a myosin type system, but the nice thing about it is that there are mutations that makes it go backward, and there are mutations that make it stop working. So again, we used this free energy calculation. Unfortunately, this is the wrong slide because they did not let me to bring my computer. But we have a very nice slide for the different mutants which reproduce this amazing effect. You see here, there are mutants that go even in the opposite direction, and there are those which just don't move anywhere. So this is what physically based approach could do. And also with this physically based approach, we found that the barrier is correlated with the protein stability. When it's more stable, it's harder to release the ADP, and therefore the ADP step is still rate limiting. Now, here comes the surprise. The guy who does the maximum entropy collaborated with the person who worked on the myosin, and amazingly, they found very nice correlation between maximum entropy to the velocity, and later between maximum entropy to stability. So basically, we have a case where the physically based approach gave you the explanation, and the maximum entropy gave you the correlation, except that it was done where we first done the physically based approach. So I think this is kind of promising for maximum entropy. And I will finish by jumping this. This is design of covalent inhibitors. But what I want to say is that we are competing with our physically based approach with AI, different deep learning approach, where people try to find a correlation. We believe, however, that combining AI with physically based approach could be the direction of the future. Of course, a bleak future will be where we will correlate everything and have no understanding, which is not good for scientists like us who want to understand why. Essentially, we were always trained to believe that understanding is the key to progress, but hopefully understanding still has major place in science. So thank you very much.